Now let's turn to our clinical candidate in the painless platform, ACD 440. So my name is Johan Sandin, and I'm a co-founder and also the Chief Scientific Officer here at AlzeCure Pharma. So ACD 440 targets a biological mechanism known as the TRPV1, also known as vanilloid receptor 1 or VR1 for short. This is a protein that plays a central role in pain signaling. And as you can see here on the illustration to the right, it is placed in a central spot to be activated or sensitized by a number of different factors, which include temperature, heat, low pH, factors or molecules that are found within the body, such as nerve growth factor, as we heard Pontus talk about, inflammatory mediators such as bradykinin, prostaglandins, etc., but also factors from outside. Most well-known probably being capsaicin, which is the active ingredient in chili peppers. And if you've ever chopped chili peppers, got it on your fingers, and accidentally put that in your eye or in your mouth, you have experienced the type of intensive pain that activation of VR1 receptors actually cause. Now, the VR1 receptor is located in the cell membrane, as can be shown here on the illustration to the right, and it's widely distributed in the body, which includes skin, mucosal tissues, sensory nerve fibers, but also in the eye and blood vessels. Interestingly, the VR1 receptor is upregulated in pathological conditions, such as in neuropathic pain patients, making this an indication that is of high interest for this biological mechanism. Now, if we look at ACD 440, this asset was in-licensed in January 2020, and it's a small molecule compound acting to block VR1 signaling. So it's a VR1 antagonist that was originally in development as an oral drug for nociceptive pain, or more specifically, osteoarthritis. The compound has been into clinical studies confirming its mode of action. And interestingly, there are also data showing from preclinical studies that it has analgesic effects in models of neuropathic pain. Now, these data, combined with the fact that you have this upregulation of VR1 receptors in neuropathic pain patients, you have a localization also in the skin, and the physicochemical properties of the compound being suitable for a topical formulation, led us to the development of a topical gel for the phase 1b proof of mechanism study with the aim to target peripheral neuropathic pain. We have also had the advantage of having large quantities of synthesized compound available for the clinical studies that came with the in-licensing deal. Now, going back to pain as an area. Now, we heard Märta talk about these three different areas of pain. You have the nociceptive, the neuropathic, and the primary nociplastic pain. As I mentioned here, we are focusing primarily on neuropathic pain, where, as we heard, this is an area with a huge unmet medical need. The majority is poorly treated, and still 7 to 8% of all adults have pain with neuropathic pain characteristics. The standard of care have poor efficacy in these patients and leading to really poor quality of life in a lot of patients. As we heard, 17% rated their quality of life scores equivalent and worse than death. So this is something that has a significant impact on daily living. Patients often show hypersensitivity. So that's an increased sensitivity, and even a normal brush may be actually painful for these patients. So this will add substantially to the ongoing pain, both in intensity and in time. With ACD440, we're targeting peripheral neuropathic pain, and we're aiming for patients with hypersensitivity, i.e. increased sensitivity to sensory stimulation, which includes patients with postoperative or post-traumatic neuropathy, but as well as painful polyneuropathy or postherpetic neuralgia. And upregulated VR1 receptors can be present in all of these etiologies. And actually this way of stratifying the patients or patient selection based on their sensitivity profiles or sensory profiles is something that's been the focus of an IMI group at EU, which was where Märta actually played a leading role, where they've developed kind of sample size recommendations and recommendations moving forward with this type of patient selection criteria. And this was also published in the highly reviewed journal called Pain. Now, there are also other indications possible for ACD440. There is clearly evidence that VR1 plays a key role also in nociceptive pain, and we have preclinical data with 440 showing potent effects in several models of nociceptive pain. So there is a possibility or a potential to also widen the indication scope for this molecule. Now, if we then move to the phase 1b proof of mechanism study that was recently communicated. This was a study that we initiated according to plan in December last year. This was a prospective, double-blind, randomized, placebo-controlled study where we looked at both safety, efficacy, and pharmacokinetics, which is looking at the exposure of the compound using the ACD440 gel. All 24 subjects were subjected to all treatments, so all subjects had both placebo and compound, so they were their own controls, if you will. We were assessing 3 different skin conditions to look at the different types of skin that the compound would encounter in the patient trials. So both looking at normal skin, so looking at tolerability and efficacy of the drug on normal skin, stripped skin, which is where you put a tape on the skin and remove that, which removes the outer cell layer of the skin, assessing the tolerability and efficacy on skin with an impaired barrier. We also had skin exposed to a light sunburn using UVB radiation to look at tolerability efficacy on inflamed skin. We also conducted a separate study where we measured the plasma concentration of the drug of ACD440 in 8 subjects with larger areas treated, and that study is going to read out within the month. Now turning to the methodology that we used in the POM study. So all subjects had 6 predefined areas specified on their backs that were either UVB irradiated, so inflamed skin, or stripped or normal skin. And they were treated with either placebo or compound, and obviously blinded to both the investigator and the subject. The subject were then put in front of a laser, which was focused on these specific areas, which evoked pain. Now, this pain was then measured in 2 ways. Both looking at pain signals using brain waves, EEG, but the subjects were also asked to assess their pain using a rating scale. Moreover, we looked at hypersensitivity, mechanical hypersensitivity, and the amount of pressure required to induce pain in these specific areas using something similar like a pinprick test. And again, the patients were asked to rate their pain. Now looking at the study results, which we communicated on April 19, the study execution was in full alignment with the time plan. So we did not have any delays due to the ongoing COVID-19 pandemic, which was obviously very good news to us. We were able to conduct the study. We did not have any dropouts. We did not have any safety adverse events, showing that the compound was well tolerated in the subjects and in this study with this topical type of formulation. The top-line data that we communicated on April 19 showed that we had highly significant analgesic effect with ACD440, both looking at laser-evoked pain and mechanical sensitivity, so on both of these different types of endpoints. Interestingly, we saw this highly significant effect both when we looked at the EEGs, that is the brain waves, as well as when we looked at the subject's own ratings of pain. Moreover, we also saw significant effects in all of the 3 different skin conditions, which is also, I think, a key step for us moving forward into the next phase. Plasma concentration analysis is currently ongoing, as I mentioned. We will get that during the coming month. Now, for the next steps with ACD440, we are now preparing for the phase 2A study that's currently ongoing. We have initiated production of the study drug. As I mentioned, we already have compounds synthesized, so it's actually production of the topical gel that we're conducting for the phase 2A study. We plan to apply for a pre-IND meeting with the FDA prior to the phase 2A study. We also aim to submit the clinical trial application, as Martin mentioned, by the end of the year. And we are working closely with our scientific advisory board of international experts in the neuropathic pain field to optimize the clinical program for ACD440. And we're also developing the full clinical development plan for the way to the market. Now, if we turn to the market, and we see ACD440 having a significant market opportunity. As we heard Märta talk about before, there is a high unmet medical need in the neuropathic pain area, so even small market shares could translate into a significant business opportunity. We have the opioid epidemic that we heard about before. This is really put the focus on non-opioid approaches in the area, avoiding the side effects and the problem with abuse and addiction. There is also a growing patient base that will provide a total market growth. And we see that ACD440 has the potential to be first in class and also has the potential placed as a first-line treatment. There is also a possibility to have as an add-on to orals, and we see this market being large, and there are not many direct competitors on the market or in the pipeline. So to summarize, ACD440 is an antagonist of the VR1, which is a mechanism that's central in pain signaling. There is also an upregulation of VR1 receptors in neuropathic pain, which has kind of moved us towards the direction of our primary indication, which is peripheral neuropathic pain, which is an area of huge unmet medical need that we heard about before. We recently conducted the proof of mechanism phase 1b study, which showed highly significant analgesic effects with no tolerability issues. And we're now planning the phase 2a study and plan to submit the clinical trial application by the end of the year. Thank you.